Hello everybody, this is Ritvik Surya and my presentation, let me just share the screen. So my presentation is on this paper by Milkens et al., uh, Professor Dr. Chad Milkens. And this is not the title of the paper, the one that you can see on the screen right now. So this isn't the title of the paper. This is a topic because I will eventually explain why this presentation is more than just a presentation of a single paper. It is more of a conceptual understanding involving me reading a lot of other related papers. So to begin with, this over here is George Ratman, and he is a Princeton graduate. And the reason why I'm starting with George Ratman is because, like our author Chad Milkens, in fact, much before Milkens, who came up to the field, George Ratman was known for his interdisciplinary work. He was actually a chemist who contributed mostly to biotechnology, and he had his own, industries. He was the founder and CEO of Amgen. He was the founder and CEO of Ecos and a lot of other industries. He was more or less an industrialist back in 1940s and 1960s. But the reason why I spoke about George Ratman is because this is Professor Dr. Chad Merkins from Northwestern University, and he was given the title the George Ratman Chemist. He was called the George Ratman Professor of Chemistry because of his contribution being mostly interdisciplinary and involving work in the field of chemistry and nanotechnology, which had immense amount of applications in medicine and biology and biotechnology. And he was, in fact, a professor of chemistry, medicine, biomedical sciences, engineering, material sciences, and nanotechnology in Northwestern University. This is the Northwestern University campus. He was also the founder of two of the university-funded centers, which is International Institute for Nanotechnology. This was a center, a research center funded by Northwestern University, and this over here is a Center for Nanofabrication and Self-Assembly, which was also funded by the university. And coming to his work, in the recent era, his work has been mostly focused on self-assembly. That's probably because he discovered his interest in self-assembly and his skills at a very early age back in 1996 with the paper which I'm going to discuss being his pioneering paper. But before that, he has explored a lot of areas in nanotechnology, including lithography and nanoparticle synthesis, fabrication, electrodes, and solid state electrolytes. Most of the stuff that we've studied, he has found applications to it, and he holds a lot of patents. So, a little bit about his background. He did his PhD in Penn State University. That's the campus. And he did his postdoc in, and I guess everyone can recognize the campus, that's MIT. And he was Dr. Milken's postdoc guide, Professor Mark Wrighton. And with him, his postdoc research was titled Microelectrode Devices for Electrocatalysis. So you can actually see how he was exploring all the fields before he came to self-assembly of nanoparticles. This was back in 1989. This was in his postdoc work was in 1991. Immediately after this, he became a professor of Northwestern University, and much before his discovery of the self-assembly, he was well popular among his peers for this discovery of a dip pen nanolithography. So this was a tool which he also got patented. We have studied lithography to a large extent in our academics. So it's basically dip pen lithography. The best analogy for that is a quill or a pen that you dip and write on. So you have a bottle of ink, and then you have a quill or a pen, and then you dip it into the ink, and then you have a surface on which you write. So the ink has Now this ink, in our case, is actually nanoparticle ink. The pen is the tool which he invented and patented, the dip pen lithography. And then these surfaces on which you write, the secondary surface on which the nanoparticles actually grow, right? In the template, or the template being the stuff that you're writing on the surface. This could be something like a gold surface or a silver nano surface, nano layers, and stuff like that. So this gave him a lot of popularity. He was actually well known for his patents and work way before his papers. And these weren't actually conventional papers. They weren't. I mean, he had written a paper, but it was not the kind of paper which goes for citation. This was more of a paper meant for patenting. And even to this day, he has work on lithography. And yeah, because of his interdisciplinary nature, he has acquired all the three titles, which is Engineering, Medicine, and Science Research Council in National Academy of Sciences. And there are several articles. This is not the only one. There are several articles where he is actually given the title the highest annually cited chemist, or the most influential chemist of his time. Although even right now it is, he is actually ruling the world of applied nanotechnology and self-assembly and nanofabrication. And he also holds a very high citation index of about 160,000 citations currently, and a good H index and I-10 index. Now, what is interesting for me here is the huge stark difference between his citations and his H index, which kind of indicates to us that his citations are unevenly distributed. One of his paper has a lot of citations, which is giving him a good number, but his H index is, it's not low. It's a very good H index, but it is somewhat, it's not balancing with his I-10 index and citations. And that is actually true, because the paper which I'm going to talk about, that is a very fundamental paper which has gained a lot of citations. And after that, his work has been more or less a derivative or an applied, areas of his own work on self-assembly. So he has been exploring his own areas with more applications. He has been giving his own citation. He's been contributing his self citations, and that is also the reason why a lot of citation is actually generated from his fundamental papers, which were back in 1990s. And even to this day, all these citations that you can see, these are from his old papers, not from the recent ones. In fact, it is very evident if you look at his homepage on his website, from the Center of Nanotechnology, and you will actually see that the papers which are featured in the very cover of the web page, it is all from those 1990s. And the newer papers are never featured. They're just cited as the ongoing work. And he has also reduced his contribution. These days, he has a huge team of nanotechnologists who work on his ideas. And he more or less works with industries and patents, and he gives guest lectures and stuff like that. So that is the bias in his citations that I wanted to bring your attention to. These are some of the awards and honors he has got. There are a lot. And some of the content that I spoke about just now is actually derived from this video lecture he gave, a guest talk he gave back in 2007, if I'm not wrong. And this is about an hour video. This is the title of the video if you want to YouTube it. And I highly recommend watching this. So he actually summarizes all of his work on nanotechnology in this one video. Summarizes in the sense he actually connects all his work, and he gives a brief biopic of his research life. These are his contact details. And this is the paper I'm going to talk about. So the first thing that should come to our attention is that this is a short communication. The paper itself is about 3 pages long. And trust me, this is actually more challenging than full-length papers, because the thing about short communications are that it is very thrilling and very bold attempt, because the author is very less defensive. He doesn't defend his ideas with a lot of references and citations. He would just propose his ideas, primarily including methodology and results and discussion, and there are no other deviations to support his arguments, which is also the reason why these papers often have what we call breakthrough ideas. But then these short communications lack interpretation to a large extent. Interpretation in terms of applicability, relevance, previous background research, those are hardly mentioned, which forces the readers to go through all the back references. In fact, I myself went through all of these are the only references which he has on his paper, just 24 references. I had, if not reading from beginning to end, I had to actually go through all these references to make sense of what he was saying, to bring in some relevance, and also look at the recent papers which he has cited. And interestingly, even in 2021, majority of his citations are from this paper back in 1996. The reason for that being, this is a very fundamental paper. This is, in fact, a pioneering paper in DNA-based, self-assembly. And after this, he has himself cited his own paper a lot of times when he has used different core materials to reproduce his work. And in fact, there has also been, if I'm not wrong, three reproduction of the same work. The exact same work has been reproduced by other scientists from across the world as a validation or some minor optimization. But that is the case. So even he is still exploring this paper. And one consequence of that is his urge to share this revolutionary idea to the world, which makes him draft his paper very quickly. And that urge to publish a paper is visible because he hardly gives any background information, he hardly cites any other papers. Uh, in fact, it looks like the entire paper has been drafted in a rush because he is not done it with his work. This was just one step for him, and he knows that after 1996 he will be publishing at least for another decade or two. He'll be just using this as a foundation to base his entire career, which is the reason why he hardly spends any time. I'm making this assumption based on reading the paper. I feel that there is a rush in making this manuscript. And having said that, this is another interesting thing which I found in the same journal. If you actually take the entire, Nature volume 382, which is the volume in which his paper was published, at the very end, you will actually see under news and views, there is analysis, a critical commentary on his own paper by Bethel et al. And it is very rare in scientific community that you actually find a commentary in the same issue. I mean, this actually shows that people are interested in his work even before the work is published. He's already in a spotlight, and let me tell you, this actually helps a lot, in a scientist's career, and it's a very rare thing. And also, this commentary is basically a sugar-coated appreciation of his paper, which means people really want to push this work ahead. Now, with my personal understanding, one interpretation I brought from this is probably because back in 1996, our human genome project was in full pace within the community of biotechnologists. And this was also the time when very newly PCR was invented, polymerase chain reaction. So probably this invention of PCR, I mean, people were actually playing around with PCR like toys. I mean, literally every molecular biologist and genetic engineers were, amazed by this wonder instrument, PCR, and Human Genome Project. So people had nucleotide synthesis at the tip of their fingers. This is what probably gave a huge growth sprout to Milkens paper, which is actually purely based on nucleotide synthesis. It involves, I mean, a great deal of his work involves nucleotide synthesis, and probably one reason why it gained a lot of popularity, attention, and respect on the day of its publication was the situation which was then prevailing. And now, this slide over here, I wanted to convey this slide because his work, the self-assembly, the concept of self-assembly itself, mainly relies on property size relation. In fact, we all have studied that size directly, causes some change in property, or the property is dependent on size. If you exploit that to the greatest possible, instance imaginable, you can actually incrementally increase the size, as in you're assembling nanoparticles one after the other, and with every single stage of assembly, from one level to another, to an upper level of assembly, you will see a different property. And this is a incremental growth. It is not like a property at point A, a property at point B. There is actually an increment in this property based on the size, because you're assembling it step by step and the entire assembly is controlled. And this property size relation was first given by Michael Faraday. So in fact, even in Milkens' paper, he actually, one of the papers which he cites is surprisingly the 1856 talk by Michael Faraday. This is one thing which Milkens doesn't forget to cite in his own paper. And yeah, so coming to self-assembly, one of the direct consequences of self-assembly in present time is the formation of superlattices. Superlattices are basically alternate layers of different materials, nanomaterials, and their assembly or their synthesis, the synthesis of superlattice has to happen by self-assembly, because you cannot allow a material to grow, because a crystal growth will be to a large extent homogeneous, right? If you're having different materials which make a super lattice, a combined lattice, then it has to assemble, it cannot grow. So that is one direct consequence of self-assembly, and it holds a lot of applications, which I will not be talking about in my presentation due to lack of time. And also the concept of self-assembly involves interactions which are non-conventional, interactions which we don't see in nanoparticle growth or material growth. That is, interactions such as van der Waals, metal ligand, biomolecular interactions, hybridizations, etc. And they are also controlled by temperature and pH and all these physicochemical conditions. And an interesting thing about these self-assembled materials is that these nanoparticles, they remain as nanoparticles when they are assembled. In case of growth, you have one nanoparticle which converts into a bulk state matter upon growing. But in self-assembly, you have multiple nanoparticles which are free, they're just particles. Then they assemble together, but they still retain their characteristic of a nanoparticle. They still exist as individual nanoparticles which are assembled. They do not exist as a macroscopic bulk state material. And so this can be visualized in this way. So if this is my nanoparticle, a bulk state of that would be just a growth. But a self-assembly would actually give rise to a framework with some periodicity, some recurrence, some geometric symmetry, and some recurring properties which are controlled by the extent of framework growth or extent of framework assembly. And there are a lot of merits to self-assembly, such as the superlattice synthesis, which I spoke about, and the profound control over properties, because I told you there's an increment from size, unlike other assemblies. But the demerits or the difficulties with handling self-assemblies, one thing is it's a poor, the long range order is poor. So this kind of framework, you don't see it for a long range. To a larger, once the assembly has happened to a certain extent, this orderly assembly will be ruined. Unlike crystal lattices, for instance, which grow to a macroscopic level and still remain ordered. And also the chemical interactions we spoke about are not adequate or defined. And before Merkin's work, this was the pre-existing ideas of self-assembly. There was use of something called a linker to assemble, to attach two nanoparticles. If I have nanoparticle A and B, which are basically the same species, they're attached by a linker. And these linkers could be organic compounds with maybe two ends of carboxylic acids, or some reactive ends which can attach to nanoparticle A and nanoparticle B. And but there is an error with, I mean, there's a problem with this kind of assembly. If you think about it, this is not reversible. It cannot be broken and rejoined and any of that. And there is prone to errors, because we can have different configurations of nanoparticles, binding to or interacting with the two ends. And also, this is not specific. We can have two of the linkers binding to each other without the nanoparticle coming in between. Stuff like that can happen. And also this growth is not controlled, it's not in our hand. Once you add the linker to a mixture of nanoparticle, you will end up getting a framework. You cannot stop the framework at say 50% or 20%. You can't really limit these reagents. So to resolve those, it is important to understand the idea of spherical DNA. And the idea of spherical DNA also was pioneered by Milkens himself. So to explain spherical DNA, these are the properties, by the way, but to explain it, I will actually take a different slide. And here I go. 1 second. Okay. So we know that double-stranded DNA exists as a linear molecule, which is one-dimensional, and then you make a loop out of it. You end up getting a plasmid, and plasmid can be called a two-dimensional structure. But then there is also one property where these linear strands of double strands of DNA, right? These can couple in such a way that they actually form a three-dimensional sphere. This is not a circle. Try to imagine that these are spheres with, it's almost like those coronas, right? You have a core and then you have these coronas coming all the way from across the 360 degrees. It's such a spherical structure. And this could happen because these ends or the terminal, one of the terminal ends of the DNA, either 5 prime or 3 prime, may have certain triggering compound which is causing the assembly to happen. Or there could be some nanoparticle code as used by our Milkens. This nanoparticle code, for instance, let us say it's gold nanoparticle code. Then if I have, say, a thiol group attached to the 3 prime end of my DNA. Then I have another thiol group to the 3 prime end of my DNA, and so on and so forth. Now, all these DNA, the thiol groups tend to bind with gold, and you end up getting such a spherical DNA structure. And these are very densely packed because this DNA strands are all negatively charged and they have a repulsion between them. So the most optimal way for them to attach is radially coming outside. And since it's in excess and there's also a salt solution like NaCl in the mixture, they end up packing the entire nanoparticle and they form a very thick, dense, corona across the nanoparticle core. And that is why you end up getting a highly dense structure. And this need not be spherical. In fact, the shape depends on the nanoparticle type. So if a nanoparticle is, for instance, a rod, right, then maybe your three-dimensional DNA of that could just be a cylinder. So you can see that it's clearly coming out to be a cylinder, right? So that way, if it's a prism, then it will be a pyramid or something like that. So this is how it works. And these cores, the nanoparticle cores, it can be inert metals or noble metals such as gold and silver and platinum, or it could be semiconductors. Or these days, there is also some amount of work happening in drug delivery system using these, such assemblies, where they use some biocompatible cores such as proteins or liposomes or stuff like that. Now, in Milken's paper, he has a very beautiful elucidation of. Just a second. So he has a beautiful way to explain, his work. And to explain this better, I would actually go through the assembly itself. He has taken two strands of oligonucleotides, which are not complementary or with similar sequence. Both the strands have a 3' thiol group. And since they have a 3' thiol group, like I said, they bind to gold nanoparticles. So the thiol group binds to gold nanoparticles. So you have basically a gold nanoparticle and a DNA tail, right? And both of this are mixed together in a single beaker. So this is one reaction system. It's all mixed together, but they just don't interact. Then in another beaker, you have these things called linker DNA, right? What happens in a linker DNA? You have a 5' 3' end, but there's a certain sequence which are complementary. So if you look at this segment over here, these two are complementary to each other. The moment they are complementary, and these two are present again in a separate system, but they are mixed together. Since they are present together, they complementary base pair. So you have something like, thymine with adenine, adenine with thymine, and so on and so forth. There's complementary base pairing. And then you have a stretch of sticky ends. So these are sticky ends because they are accepting some amount of, base pairing from another species. They are just sticking out with their phosphate groups and their hydrogen bond centers so that they can hydrogen bond with other nucleotides which are complementary. Now, if you take this part, and this thing would basically look like this, right? This is what a linker would look like actually, right? And these are the sticky ends. And if you feed something which is complementary to the sticky ends, then you basically have those something, this the stuff that you're feeding to the sticky ends attached this way. So in a way, this linker is basically linking these two strands of DNA, strand A and strand B, via these open sticky ends, these sticky ends over here. Now, what we do is we add this to our initial mixture, and very easily you can see that since all of these are basically synthesized, these oligonucleotides are artificially synthesized and polymerized because of PCR and chemical synthesis, which was then prevailing. You will see that there is complementarity, again complementarity. So there is a binding, and this is how the binding would proceed. So basically, what I had was one gold nanoparticle and a stretch of DNA. I had another gold nanoparticle which also had a stretch of DNA. So this was from 3 prime, and this was again from 3 prime. So you know that a double-stranded DNA is always from 3 prime to 5 prime and 5 prime to 3 prime, right? So if I have 3 prime with gold nanoparticle, then I have another 3 prime with gold nanoparticle, and I add a linker. A linker basically does this. So the end result is you have one gold nanoparticle and then you have another gold nanoparticle. Both of them are linked by this stretch of DNA and the linker. So the analogy for this would be if gold nanoparticles are atoms, then this would be a bond. The DNA would be a bond. So just like how a bond binds the atoms, the structure would look something like that, where these gold nanoparticles are bound by DNA and linkers. Now, this is what is happening in the reaction mixture as well. Again, we have these two strands fabricated with our gold nanoparticle. They have thiol groups. So since they have thiol groups and then you are adding gold nanoparticles to it, it is forming these spherical DNAs. The spherical DNA is all mixed up in a single reaction mixture. They do not bind. The moment you add the linker, they will start binding. So this is when you're adding the linker, and then you allow it to polymerize or oligomerize, and then they form this assembly. This assembly is basically a self-assembled framework. Why is that a framework? Because these particles do assemble in an orderly fashion. They assemble in a very rigid fashion, and they're all three-dimensional. They have a perfect framework. And the beauty of this is the entire process is reversible. I will tell you why it is reversible. If we talk about a spherical DNA alone, it cannot bind to each other unless there is a linker added. Why? Because these DNA are all negative, these are all negative. There is a negative negative repulsion. If it was only gold nanoparticles, they would have formed a colloid. They would have precipitated out. But since these are spherical DNAs across gold nanoparticles, they will not bind together unless you're adding a linker. So that is in your control. Once you add your linker, fine, they bind to each other and form a framework. Now, since this is by oligomerization, there is also alkaline-based denaturation of DNA, acid-based renaturation. So you decrease the pH, you end up assembling it. You increase the pH, you end up dissociating it. Also with temperature denaturation. Since we are synthesizing these oligonucleotides, there is a formula to calculate the denaturation temperature, which was found to be 42 degrees for our example sequence, which means you take a double of this. Let us consider 80 degrees and then go all the way down to 0 degrees. At 0 degrees, they would have assembled. At 80 degrees, they would have disassembled because of denaturation. So temperature or pH or all of these are like switches. You switch it on, there is dissociation. You switch it off, there is reassociation. It is perfectly under control, and this entire process, I mean, it's a very beautiful way of actually controlling this process. And this is what it looks like. Now, this is at 80 degrees Celsius. Since it's at 80 degrees Celsius, I will say that this is denaturated. You just have spherical DNAs everywhere. And what you see, this color, the red color is because of the DNA which is visible. So this is recognizable at 700 nanometer. I'm sorry, this is because of the gold nanoparticle which is visible. So 700 nanometer is the absorbance maxima for gold nanoparticles, and that is why this appears red. The moment I reduce the temperature all the way down to 0 degrees, this is actually room temperature, I drop it down to 0 degrees. What I see is there is linker present in the mixture, but the linker does not link. But at 0 degrees Celsius, the linker starts to renaturate. And as a consequence, you have assembly formation. The moment there is assembly, you can actually check this at 260 nanometer. 260 nanometer is the absorbance maxima for DNA. So here the characteristic absorbance is by DNA. Here the characteristic absorbance or the maximal absorbance is by gold nanoparticles. So if I consider, say, one of these wavelengths, let us consider 700 nanometers. Then I cycle the temperature. Since we are using a thermocycler, let us cycle it from 0 to 80, 80 to 0, 0 to 80, and so on and so forth. So what you will see is at every 80 degrees Celsius, there should be a peak at 700 nanometers. In fact, this is what was observed. At every 0 degrees Celsius, there is a peak at 700 nanometers. 0 degrees, there's a peak at 700 nanometers. Why is there a peak at 700 nanometers? Because at 0 degrees Celsius, there is renaturation that would have occurred. And because there is renaturation, this is the color which is visible. But at every 80 degrees Celsius, our 260 nanometer is showing a peak. So 260 nanometers at 80 degree. At 80 degree, there's denaturation. Since there is denaturation, DNA is the principal absorbing agent. Hence, it's showing an absorbance at 260 nanometer. When it is renaturated, again back to 700 nanometer, which is the absorbance range for gold nanoparticles. So this is a perfect example of how the assembly and this disassembly is switching, and it's very what do you say very symmetric. I mean, you just switch it on and off, and instantly, I mean, this is very instant. You can look at the peak, right? It's very, very instantaneous. The moment the temperature is dropped, there is a rapid assembly. The moment it is raised, there's a rapid disassembly, and there is no loss in the compound. In fact, all of them are assembled or disassembled in the same peak. There is no difference in concentration, so it's all happening almost in a conserved fashion. And this was some of the TEM images observed. And again, this is a comparative image of a growth. So if I have gold nanoparticles and I just allow it to grow, so I have a nanoparticle, allow it to grow, it becomes bigger. That's it, right? So this is a very typical situation of growth. But if I have the same gold nanoparticles and I allow it to assemble, it will remain as nanoparticles, like I said, but it will just form a framework. That is a very beautiful thing with a lot of potential applications, and that is visible in the TEM. You can see that they're still as nanoparticles. The only thing is they are associated, right? But here they've just started growing into bulk state material. And here again, the association is not very symmetric. Like earlier I said, it forms a framework. You can't really see that framework here. It looks more scattered. That's because it's a TEM, and you can't really see the 3D framework, right? Because TEM visualizes in a two-dimensional pattern. So you're saying a lot, what you're seeing there is a lot of overlapping. So to actually see the framework, you should be at this point able to visualize this, right? This is what a framework would look like, where these are your gold nanoparticles, and the DNA is forming something like a bond or a framework or a foundation holding all the gold nanoparticles in place and showing a relevant property. And the whole thing being customizable, and there is a gradient of temperatures or pH where there is a gradient in the assembly, and everything is under your control and reversible. So having said all that, there is a lot of applications, but I would unfortunately not be able to explain those applications in this presentation because of lack of time, and also because all of this was not involved in Milkens' paper, which I refer to, the 1996 paper. He has, like I said, published a lot of papers much later where he has explained all these applications. So, for example, the spherical part, the radiating coronas, need not just be DNA. It could be peptides, it could be receptors, small molecules, antibodies. Now, all these could, again, because all these can still hold nanoparticles, it can still hold, you can still bind to each other. Receptors can bind to each other, like just like linker DNA, and it can still form a framework of nanoparticles. And also the core itself, nanoparticle, it need not necessarily be nanoparticles. This could be more of biologically compatible compounds, but then again we are going away from nanotechnology there. But that is also the reason why it has a huge potential application in drug distribution and drug delivery. But none of this work was actually cited by Milkens. These are all consequence of the work in present time. Again, the slide is pretty self-explanatory, but I won't be able to explain gene regulation using this technique of self-assembly because of lack of time and lack of relevance to Merkin's paper of 1996. And yeah, more applications on other different kinds of growth that can happen. So need not just be gold frameworks. Now you can see that there's also a cesium chloride framework, NaCl frameworks, all because of DNA aided framework. It's a very popularly explored field in the current nanotechnology. And these are some of the other papers which I referred to. Apart from Milken's work, I have also referred to these papers and all the papers which were cited in Milken's paper, which, if I'm not wrong, I have explained in a previous slide back in the very beginning. So, yes. So, these are all the papers which I have referred to. And this presentation was not with the sole paper of Milken's because of the fact being that Milken's paper itself is short communication. So with that, I would like to conclude my presentation. Thank you so much for listening.